Hello, everyone. I'm Yansha Deng, Senior Lecturer from King's College London. Today, my topic is chemical reaction-based microfluidic circuits for molecular communication design, analyze, and prototype. So in this talk, I will present the background and motivations, and then introduce the design, analyze, and the prototype of our chemical reaction-based microfluidic circuits, and finally, the conclusion. Communication technologies for transmitting information over distance have been revolutionized the way human sensors, vehicles, and robots communicate from 1G to 6G. They are information carriers emitting EM waves, which are insufficient for nanoscale and microscale applications, such as drug delivery, chemical sensing, etc. So therefore, there is a need to create an efficient and reliable communication paradigm at nano or micro scale. Different from existing wireless communication systems that are designed for typical human-to-human, human-to-machine, and machine-to-machine microscale communications, molecular communication is proposed to transmit chemical signals such as molecules and achieve human-to-environment and even to human-to-the-cell and cell-to-cell communication at extremely small dimensions, such as micro or nanoscale dimensions. And in this way, mimicking the biological communication in the nature. To explore molecular communication signaling in cell biology, jointly with Adam and Robert's group, we propose a 5-level communication hierarchy to be mapped directly to the biological behaviors, with neuron communication as an example here. Level 1 defines how molecules are propagated and transported between the transmitter and the receiver after they are releasing from the transmitter membrane and before they interact with the surface of the receiver, like the diffusion of neuron transmitters over synaptic cliff. Level 2 defines how the physical signal is generated at the transmitter and sampled at the receiver, like the generation, release, and binding of the neuron transmitters. In our early works, we studied the modeling and the simulation of molecule generation and molecule reception by reversible absorbing receivers in our paper listed here. Level 3 defines the conversion from data to a physical signal and observing a physical signal to recover the data between the microscale and nanoscale. This we call modulation and demodulation here. From the biological perspective, this level also includes observing and controlling the microscale change using a macroscale equipment in the lab shown on the left and right of this plot. This establishes the interface from the micro world to the micro world for molecular communication. For example, we can use pumps to release the chemicals to change the pH of a microsystem and rely on a pH measurement instrument to observe the resulting response. And we leverage this example for our testbed in the later slides. Level 4 defines how the information is in the concentration signals is processed within the transmitter or receiver, which include various analog and digital signal processing functions for chemicals concentration signaling via genetic circuits and chemical circuits. Genetic circuits rely on synthetic gene networks to produce desired responses, while chemical circuits employ a set of chemical reactions for targeted response. Level 5 defines the more competitive collaborative behavior interactions between the molecular communication device for diverse application which requires multidisciplinary collaborations. Today's presentation mainly focuses on how to manipulate and process the chemical signal from the level 1 to the level 4 in a molecular communication testbed. Existing molecular communication testbed, either gas or liquid-based, have been implemented to demonstrate the feasibility of exchanging information, while alcohol molecules, DNA molecules, magnetic nanoparticles, fluorescence, acid or base, or other molecules, etc. However, these chemical concentration signals are first measured through mechanic and readout device, such as pH sensor, alcohol sensor, mass spectrometer, and then leaving the rest of the analog and digital signal processing to the external electrical controllers, such as Arduino or laptop, based on electrical signals. They can hardly support the biocapable, non-invasive, and microscale and nanoscale requirements of biomedical applications. This motivates us to design molecular communication components capable of processing and exchanging information over molecule domains to unleash interdisciplinary applications. Recall that signal processing over molecule domain can be realized via genetic circuits or chemical circuits, and have enormous genetic circuits has an enormous advantage, but it faces challenges such as low speed and unreliability. So this motivates us to start by designing chemical circuits, which have advantages in rapid prototyping and easy control. Notice we also have ongoing works on designing and analysis of genetic circuits that can be found online. In particular, we are interested in the liquid-based chemical circuits because the likelihood of the chemical reactions over the air can be limited substantially by the large mean free path of airborne molecules. Whereas a liquid-based system on the right allows for a large variety of reactions with kinetics and a higher likelihood of reactions. Indeed, liquid transceivers are not new in communication engineering. One example is a gas liquid transmitter that relies on liquids to transduce sound waves to electrical currents. Of course, none of the existing liquid transceivers relies on chemical reaction for their operations. In our research, we are particularly interested in the integration of chemical reactions and microfluidic devices so that we can jointly control the occurrence of chemical reaction following a sequential order while the flow inside molecular communication transceivers through processing small amounts of fluids in dimension of micrometers. Next, I will first introduce our progress in the design and analysis of microfluidic circuits. Our first contribution is that we propose a 5-layer architecture as an abstraction of a microfluidic processing system to facilitate microfluidic circuit design. As shown in this figure, we mapped our proposed layers of microfluidic processing system to those of electronic processing system on the left. In the layer 1, the movement of the chemical particles in the microfluidic system is mapped to the propagation of electrons in the electrical system. In layer 2, the reactions for signal transformation are mapped to the transistors. In the layer 3, the basic modules perform simple arithmetic calculations such as addition, subtraction, and amplification. Operations in the microfluidic system are mapped to the analog circuits in the electronic system. In the layer 4, the microfluidic logic gates are mapped to the electrical logic gates for digital, chemical, and the electrical signal processing respectively. In the layer 5, complex microfluidic or electronic circuits can be constructed to achieve a specific signal processing functions, such as modulation, demodulation, encoding, and decoding for molecular communication system. Based on this novel system architecture, we designed several basic microfluidic components from layers 1 to 3. For layer 1, we presented 3 basic microfluidic geometries for molecular propagation, including Y-junction, T-junction, and straight channel. For the layer 2, the chemical reactions are introduced into advection diffusion channels. By introducing the chemical reactions, the microfluidics are in those with the signal processing capabilities. And by combining the components, designing layers 1 and layer 2, we can construct the addition, subtraction, amplification modules in layer 3. Based on the modules designed in layer 3, we apply them to construct AND or XOR gates for layer 4. The realization of signal processing function while microfluidic circuits show significant advantages in is versatility and modularity. The versatility here means that it is reflected by the AND and OR gates sharing the same microfluidic geometry structure, but with different reactions and species concentration. The modularity here is that our designed microfluidic logic gates can be constructed while a combination of 3 microfluidic modules, which is similar as to the Legos that can be plugged and played. With versatility and modularity, more complex signal processing circuits at layer 5 are envisioned to be built through a combination of logical gates at layer 4. In the next slides, I will present one example of layer 5, which is binary concentration shift keying, we call BCSK, modulation and demodulation, microfluidic circuits, building from a combination of logical gates at layer 4. In our design, the BCSK transmitter is capable of generating a predefined signals upon the transmission of bit 1 with a rectangular shape. And the chemical reactions are directly inspired a network motif. It's called an incoherent one feed-forward loop in biology. The amplitude of the transmitted signal can also be adjusted by changing the transmitter geometry. And here the figure shows the simulated result obtained from the COMSOL simulator at the output of the transmitter. After the signal propagation through the molecule propagation channel, at the BCSK receiver, we designed a thresholding reaction and amplifying reaction. And the BCSK receiver can demodulate this pulse back to a rectangular signals. More detailed modeling, analysis, and simulations are provided in our best paper and journal version. And we also designed a more complex QCSK function that constructed using layer 4 and validated in our recent accepted TCOM paper. So far, I have many focus on the design, analysis, and the simulation of microfluidic circuits. However, there are still practical implementation issues. It faces the flow chemistry challenge on the microfluidic geometry design and the chemical molecule selection, and also facing the communication engineering challenge on the control and the optimization of the communication efficiency. So therefore, it is essential to develop a proof of concept platform for chemical reaction-based microfluidic circuits. Now I will introduce our chemical reaction-based microfluidic circuits prototype. Like any communication system, our experimental MIMIC platform is composed of 3 unit blocks connected to each other, a transmitter, a propagation channel, and a receiver. The transmitter of our platform includes 2 functions, which are signal generation and signal shaping. Signal generation function is achieved by using a syringe pump to carry a liquid phase signal. This pump converts input electrical signal to a variation of the concentration of the chemical Y using a simplified BCSK function. The injected signal Y first reach the mixed junction located inside the transmitter, where it can react with the signal repressor P. By controlling the timing of the injection of P, we can control the time at which Y is consumed by the reaction. And then this will design the final shape of the signal Y. This allows us to convert a constant injection of chemical Y into a squared pulse signal entirely through the reaction between the Y and the P. The shaped signal is then sent to the receiver via the propagation channel. In our platform, the propagation channel is a microfluidic tubing, which length can vary depending on our needs for each experiment. In this study, we tested up to 25 meters of tubing to propagate the signal in Y from the transmitter to the receiver, with a certain distortion of the signal. After propagating and upon reception of the signal Y at the receiver, we introduced the thresholding and amplification reactions for signal detection. We first designed a thresholding operation to mitigate the noise and the inter-symbol interference introduced by propagation channel. Through the reaction between the Y and the signal thresholder THL, the reaction effectively consumes all the signal Y below a predefined concentration level of threshold. To amplify the remaining signal Y to a detectable level, we then devise the amplification reactions to generate a receiver output and a signal amplification amp to generate a receiver output O, as shown in the figure. It is essential that the output O is visible to the detector connected to the receiver. And here we use ultraviolet visible spectrometer due to its versatility. For our proof of concept experiment, we selected sodium hydroxide NaOH as our signal carrier Y and encoded the signal in a variation of concentration of NaOH. We then select the hydrochloric acid HCl as both our signal suppressor P and the signal thresholding THL. We select the solution of the BTB as the signal amplifier, and the BTB is pH sensitive dye, a chemical whose main property is that its color will change with the pH of the solution. Specifically, the pH is characterized by the acid form of low pH value and the acid form called HPTP, yellow in color, and then a base form at high pH value called BTB minus, blue in color. In the presence of Y, the yellow from the BTP can be transferred to a blue form. And in this way, we can show the signal appearance. Assembling all these blocks and functions leads to a full prototype in these slides. The transmitter is controlled by Arduino that controls 3 pumps injecting Y, solvent, P species with their selected chemicals for the signal shaping functions at the transmitter. After the signal propagation, the received Y signal will be reacting with the thresholding and amplification species with their selected chemicals, and then based on the signal thresholding and the signal amplification reactions at the receiver. And these are measured by a UV spectrometer. Under this design, we were able to build our prototype in the lab as shown in these slides. We can see here the transmitter is at the top level of the structure connected to the propagation channel, shown here in the back, and leading to the receiver occupying the bottom level of the structure. We also highlight the location of the UV spectrometer used for the detection of the signal. The spectrometer can be found here in the back of the platform, and it reads the signal in the solution following through the flow cell, connect to the output of the receiver, and is powered by the light source here. All these components composing the receiver of the platform are connected to each other using the optical fiber. All the platform is operated using a Python software we developed ourselves. So the Python software operates all the pumps of the platform as well as the spectrometer to read the signals in real time. To ensure the flow stability, flow meters are connected to the syringe pump, and our software integrates a feedback loop through a PID controller. Moreover, this software allows us to automate all the experiments and communication experiments through a pump scheduler function. The software will be released and made available under the open source license. And knowing that with this platform, we will be able to remotely access the platform and operate automatically when we work from home during COVID. The syringe pump on the right are designed and 3D printed by our group, and are based on the stepper motor operated by Arduino board and controlled by the Python software. Our 3D printed pumps reduce the cost of 1,500 pounds for a commercial pump to 100 pounds. With all the signal processing chemical reactions selected and set, we are able to use our Mimic platform to transmit a message between the transmitter and the receiver of our prototype. We tested our platform by sending a simple message "Hi" from the transmitter to the receiver of the platform. The different characters composing the message were first encoded into the bits with the basic ASIC conversion, with each character represented by a series of 7 bits. The electronic bits are converted into chemical signals by actuating the syringe pumps. To represent bit 0, no concentration of Y is injected during the bit interval. To represent bit 1, a solution of Y is injected during a fraction of bit interval. We call it duty cycle alpha. And for the initialization, 2 initialization bits are inserted at the beginning of the message. And this allows us to transmit a message "Hi" to using our Mimic platform. So we present here a time elapse to show exactly how the solution color change when the bit 1 is being transmitted between the transmitter and the receiver of our platform. So as you can observe, the yellow HPDB, which indicates that no Y is present in the solution, turns into a blue PDB solution upon the reception of signal Y. And this shows the presence of Y signal. Our experimental result demonstrates here that the transmitted message was successfully demodulated at the receiver, and all the bits were recovered with error-free transmission using the value of bit interval encoded in initialization pair. Note that here the long bit interval and small flow rate ensures the completeness of all the reactions in the tubing and a stable flow rate output from the syringe pump. So we first test the capability of our platform to transmit message by assessing its performance at high speed, up to the limit of our injection system and over long distance, with a propagation channel up to 25 meters long, more than 6 times longer than any propagation length previously reported in the literature. As demonstrated in our result, signals transmitted at the highest speed we could achieve, and also over long distance, we can still detect, the signal can be still detected by our platform. Moreover, the parameters of our chemical reactions can be fine-tuned to control the reliability of the communication. For instance, we demonstrated here experimentally that the duty cycle alpha set by the reaction between Y and P can be modified to impact and reduce the bit error rate. However, using chemical reactions to process the signals can introduce new source of error, and the signal distortion we have to take into account. More details of our chemical design and result are provided in the paper, which will be posted online later on. So finally, I would like to conclude that until now, we have been constantly working on the microfluidic circuit, starting from the microfluidic design for the basic 5-layer architecture, and then we designed the BCSK and QCSK realizations. And then we actually move a little bit to the prototype, where we validated that the BCSK can be deployed and used for modulation and demodulation to transmit signal message high over and even 100 bits over a large distance. And we can also optimize the communication with higher speed and high data rate. And to this end, we want to also conclude that our platform has a versatile design and modular platform that can be adapted to much variety of different chemical reactions testing in the platform, and even genetic circuits testing as well. And at the end, I would like to thank all my collaborators, especially Massimiliano Piopon. When I visit there, I get the inspiration of the idea on the chemical reaction networks and the genetic circuits. And also, I thank Professor Rob Schopper for his very helpful comments to our survey paper, and then Adam Noy for collaborating with us on the some of our research work and the survey paper, and Dr. Ali from Imperial College, with our close collaborator to build the MIMIC platform. And last, I also want to express my grateful for my two postdocs, Vivian Water and Dadi, who at King's College London, who has been actively working on this prototype in the past 1.5 years, and delivered a very nice result over this 2 years period. And this work is funded by the EPSRC New Investigator Grant as my first grant. And thank you so much for all the support of King's provided during the past few years on my research. Thank you.